Okay, so
Okay.
a warm welcome to the Kiel Imaging Seminar. It's great to have you all here today. It's my pleasure to introduce our KIS speaker to you, Professor Rachel Katz-Brull. She's a leading scientist at Hadassah Medical Organization and a professor of imaging at Hyperpolarized University, Faculty of Medicine. She directs a Center of Hyperpolarized MRI molecular imaging in Jerusalem. Her pioneering work focuses on developing advanced molecular imaging agents for MRI, including innovative hyperpolarized MRI techniques that allow for non-invasive imaging without ionizing radiation. Professor Katz-Brull's research has significant implications for diagnosing and understanding conditions such as breast cancer, liver disease, and brain metabolism. She earned her PhD from the Weizmann Institute and completed postdoctoral research at Harvard Medical School, where she made groundbreaking contributions to liver disease diagnosis through MRI spectroscopy. Before we start, to the audience, please wait with your questions until the discussion. And with this, speaker Professor Katz-Brull, the stage is yours.
Thank you very much for this kind invitation to speak at this distinguished symposium. And thank you for the introduction. So today I will talk to you about hyperpolarized MRI agents and other MRI agents as alternatives to gadolinium-based contrast agents. Gadolinium-based contrast agents are wonderful agents for medical imaging, but they may cause major health concerns as the long-term accumulation of gadolinium in patient bodies and the environment could lead to a variety of health problems. So we asked, is it possible to find alternatives to gadolinium-based contrast agents? And in order to answer this question, we formulated some principles that we'd like to adhere to. So this new class of MRI contrast agents should be metal-free, safe, and eliminated from the body and the environment. We started this project with this concept with a project that was funded by the EC, and it was named Alternatives to Gadolinium. From this project, we actually have now 3 new projects that are funded through work that was done in this project, and these are Response, Precious MRI, and DDG MRI. So I will talk today about several possible new agents for MRI. 2 that are non-metabolic agents, and these will be urea and deoxyglucose. And 1 that is a metabolic agent, and this is pyruvate. We have this project with nanodiamonds and pyruvates. On this I will talk less today, but I will talk about the possibility to use the metabolism of pyruvate as a contrast agent in MRI. So when we think about new MRI agents, whether they are hyperpolarized or not, because they are multinuclear, we have to consider and the hyperpolarized technology and the MRI detection of the multinucleus. And in hyperpolarization, we have to consider several formulations: the formulation for hyperpolarization, the compound that contains this multinucleus, and then the formulation for transfer and the formulation for injection if they are different. So I will speak about all of these aspects in the various topics. And then we have to consider the biology. First of all, the safety, the biodistribution properties, the tissue specificity, and eventually the utility. Is this agent useful at all for clinical imaging? So for hyperpolarized agents, we are looking for long T1. And in order to produce high-resolution images, we prefer to have a non-metabolic compound. So 1 candidate was N15, and we looked for compounds that contain N15 in order to see if they could serve as hyperpolarized agents that are useful in the clinics. The first one that we looked at is sodium nitrate. This is nitrate, this is the ion. It's not used in imaging at all. It is used in food and agriculture. But we thought that it may have a very long T1 based on the literature. And indeed, we found that when we keep it at colder temperatures, you see 12, around 12 Celsius, it has a long T1 of about 170 seconds. And we just couldn't check colder temperatures, but it's probably even longer at colder temperatures. So we saw that when we keep it at this cold reservoir of hyperpolarized nitrate, we could inject it again and again to blood and see the hyperpolarized compound, the high signal again and again. Then we tested its safety profile, and that, together with the literature, showed that it is very safe for bolus intravenous injection. However, the polarization that we have been able to get has been low so far. Maybe it could be improved, but so far it has been low. Another agent with the N15 that we looked at is N15 labeled urea. This is the compound. And because the exchange of protons with deuterium is so fast in water, when we dissolve it in D2O, we actually have deuterium-labeled urea. So we looked at this compound and we dissolved it in D2O. Why? Because as you can see here, at thermal equilibrium, the T1 of urea dissolved in D2O is much higher than that in H2O. Now here again, we looked at the effect of temperature, and it turns out that it is the opposite way. So for urea, when we keep it at hotter temperatures, so up to 75 degrees, we get a very long T1 of about 6 minutes. So we did the next thing, the same experiment as with nitrate. We injected this high temperature D2O reservoir of hyperpolarized urea to blood, and we can see the hyperpolarized signal again and again. So we decided to focus on this, on hyperpolarized urea in D2O. This is the exchange. And we note that we perform the polarization and the dissolution in D2O. And then we asked, what about the ratio of H2O, D2O? Because when we will inject the compound to the blood, it will be mixed with regular water, non-deuterated label. So we checked the T1 of hyperpolarized urea in various combinations of water and D2O. So as you can see, in 100% D2O, the T1 is high, but even at 50% D2O, we still have a long T1 of about 1 minute. So this was encouraging. We also checked the effect of gadolinium in the formulation on the polarization, and we found that a little bit of gadolinium in the formulation, 1.3 millimolar, was useful to get higher polarization. Then in the joint project that we had named Alternatives to GAD, the University of Cambridge was one of the partners, and they were able to significantly increase the polarization of urea. We had about 5%, but in a new device, in a new polarizer that operates at 7 Tesla and 1.2 K, they were able to produce 3 dissolutions within 12 minutes each, and the polarization percent was 40%. They were able to detect urea in vivo, in the liver, and in a brain tumor, and they were able also to transfer the polarization to protons for proton detection. In a paper that will hopefully come out soon, they have shown also that hyperpolarized urea could detect xenograft tumors in mice. So we had another question about hyperpolarized urea, and this was a more basic question of why is the polarization of urea dependent on D2O? Do we get higher polarization because nitrogen 15 is directly bound to deuterium when it's dissolved in D2O, or is it the effect of the bath, of the D2O bath in the polarization? So also this we approach within this project called Alternative to Gadolinium with the University of Oxford with Damien Tyler. And we found that actually the directly bound deuterons increase X-nuclei hyperpolarization by DNP. So first we looked at carbon-13 because the polarization is higher and it's easier to look at. And also we were working with the hypersense at the time, and there are limitations on N15 observation in solid state. So when we look at glucose that is labeled with carbon-13 and deuterium, meaning that all carbon positions are directly bound to deuterium, compared to glucose that is non-labeled with deuterium, we can see a much higher polarization. And the same we saw for 2-deoxyglucose. In D2O, the direct bonding to deuterium increased the polarization in solid state. And then we asked, maybe still there could be some bath effect. So we compared the directly bound deuterium and carbon-13 in glucose in D2O or H2O, and there was no effect of the bath. The polarization was the same within the error limits. And then we came back to nitrogen 15 in urea. So as you can see here, the polarization in D2O is much higher than in regular water, although the effect is not as dramatic as in carbon-13. And when we looked at the same thing for nitrate, which has no directly bound deuterium, even when it is dissolved in D2O, there are no protonated positions. So here again, we saw no effect of the bath. So we decided that this effect has to do with direct bonding between X-nucleus and deuterium. In short, our conclusion was that a non-metabolic agent, which is N15 urea, dissolved in D2O, could serve as an alternative to gadolinium-based contrast agents in detecting tumors specifically. And now I would like to go to another agent that we are developing right now. And this one is actually not hyperpolarized, so I changed a little bit the title of the talk. So this one is actually at thermal equilibrium, but still it contains a multinucleus, and this is deuterium. So for thermal equilibrium, we need very short T1 because we want to average fast. And we still want an agent that is non-metabolic to get good images, so high-resolution images, so that signal is not distributed among several species. So one non-metabolic agent that we propose is deuterated 2-deoxyglucose. So all of the positions are labeled with deuterium. And we claim that this could serve as a parallel to FDG PET without ionizing radiation, but this we still need to prove, but I will walk you through the rationale. So this work was inspired by the work of the Fighter et al., who showed that deuterium metabolic imaging in patients could detect tumors by lactate production in the tumor from deuterium-labeled glucose. And I like this work very much because already in '98, I liked using deuterium NMR. So this spoke to me. And I would just like to, before I go on, I would like to remind you how FDG PET works. So PET is positron emission tomography, and FDG is 2-deoxy-2-F18-D-glucose. So it is labeled with radioactive fluorine ion. And the reason it is locked in the cells is that it undergoes phosphorylation. So this radioactive molecule is locked in the cells, and because cancer cells take up a lot of glucose, these radioactive molecules serve as imaging agents for PET and for detecting cancer. This is a very important clinical imaging modality and application. However, it is not used in the general population. It's not a screening method. It's only for people with proven cancer because of the high ionizing radiation that is involved in the PET examination. So the Fighter et al. Used glucose, not deoxyglucose, and they labeled it with deuterium in position 6. So the parallel with respect to FDG PET would be a deoxyglucose that is labeled here in position 6 with deuterium. But if we label it with more deuteron atoms, then actually we will get a higher signal. So that's the rationale. Before we started, we wanted to make sure that the T1 is really short and it could allow for very fast averaging. So we checked the T1 of deuterated sites in several molecules. And we saw that when the deuterons are next to a ring or on a ring, their T1 is actually very short. So 56, 43 milliseconds. So this could allow for for a lot of averages per unit time. As an initial experiment, we carried out an experiment with deuterated glucose, like in DMI, but here we were interested in ischemic stroke, and we worked a lot with hyperpolarized compounds on ischemic stroke, and we have this model of brain slices where we can stop the flow to create ischemia. So we wanted to see what would be the response of deuterium metabolic imaging, not imaging, but spectroscopy to stroke. So as you can see, once we add labeled glucose to the perfused slices, so these are viable brain slices. They are electrically active. It's an ex vivo brain. When we add glucose, you can see that immediately lactate is formed. And when we induce ischemia, immediately lactate goes up very significantly. And upon reperfusion, lactate level goes down. And on the same slices, we induced a second ischemia, and we saw the response exactly the same. So this shows us first that the brain takes up deuterated glucose, that the time scale is very fast, because in DMI experiments, people usually wait 1 to 2 hours from administration of glucose through to the measurement. So we can actually see that the production of lactate in the brain from glucose is extremely fast. And we also compared the reduction in glucose during the ischemia periods, and it was exactly the same as the reduction in glucose in the test tube. So actually lactate indicates glucose uptake in the brain. So this is a test case, and we believe that the deoxyglucose would actually show us glucose uptake in the brain and in cancer tissue as well. I will spare you this pathway. But the main point is that from each glucose molecule, we have one labeled lactate molecule with 2 deuterons and one lactate molecule that is completely non-labeled. And just to complete the story, many years ago, we worked on hyperpolarized glucose that is labeled with carbon-13 and deuterium. And we saw nice images, and we hope that now with both hyperpolarized dual-labeled deoxyglucose or with deuterium-labeled deoxyglucose, we could get a parallel to PET. And so now I would like to talk a little bit about the metabolic agents. So hyperpolarized pyruvate has been suggested by many of you and in the world to be an agent for detecting cancer and other indications. And I would like to tell you about our contribution to understanding how pyruvate works and what affects its metabolism in various tissues. So this is actually the original aim of hyperpolarized agents, and there they needed long T1, which pyruvic acid has, and the second demand is that they do demonstrate metabolism. So now we will talk about a metabolic agent. And the first study that I would like to show you is all of the studies that I will show you are ex vivo studies, so not in vivo, and this makes them special in the way that they show only the metabolism in the investigated organ. Whereas in vivo, there could be interference from other organs or other systems. And ex vivo studies are also enable a higher degree of control of pharmacokinetic. So the first example is real-time in-cell metabolism in perfused heart and brain and the response to stress. I will just remind us what is the hyperpolarized pyruvic acid, what is the metabolism that we see. So from pyruvate, we could get lactate by LDH or alanine by alanine transaminase or bicarbonate by PDH and carbonic anhydrase. The acetyl-CoA goes to the TCA cycle, but we don't see it because it is not labeled. So the label actually that most people use enables us to watch for lactate production, alanine production, and bicarbonate production. If we want to see the TCA cycles, we have to label also on position 2. And in most of the studies that I will show you today, we will only see lactate and bicarbonate. So the first thing we looked at was the perfused heart. So you can see a mouse heart beating inside an NMR tube here. And this was the system that we worked on. So the hyperpolarized agent comes and the perfusion comes directly to the heart in a retrograde flow. So this is our system. We have the heart. It's attached to a peristaltic pump. We put it inside an NMR spectrometer. We measure phosphorus spectroscopy to make sure that we have ATP and that the pH is correct. These studies were done on a Hypersense. Now we have a spin aligner machine, but we inject hyperpolarized pyruvate, and then we look at the production of lactate and bicarbonate. I will not go into the calculation, but we get actual rates. And we work with selective pulses, so we either see lactate or bicarbonate, so one spectrum, alternating spectra. To cut the story short, we evaluate the pH of the heart tissue and compare it to LDH to PDH activities ratio. And what we saw in a group of several hearts that had a variation in pH due to probably the surgery, which is very delicate. So it's really hard to get the cannula exactly in the same place. So the pH is not the same in all hearts, and it could also vary during the experiment. So between hearts and between injections, every heart got 3 injections of hyperpolarized pyruvate, we saw that there is a clear link between LDH to PDH and the pH. So the lower the pH, the higher LDH to PDH activities ratio. And we wanted to understand more if this is always true or not always true, and what is the mechanism behind this. Is it the pH that affects LDH and PDH, or is it the other way around? And to answer this question, we also used inhibition of the main pH regulator, the sodium proton exchanger. So this is a cardiomyocyte, and LDH activity occurs in the cytosol, and PDH activity occurs in the mitochondria. So intracellular pH could affect LDH, maybe it could affect also PDH. So this is the question that we wanted to answer. What is the effect of intracellular pH on LDH and PDH activities? There is a clinical context to this question because many clinical conditions have accompanying extracellular pH reduction or acidification and intracellular acidification. And therefore, when we look with hyperpolarized MRI on LDH and PDH activities, we have to take this into consideration if there is an effect, as we saw in the previous work, to interpret the clinical imaging correctly. So we used 2 intervention tools to control intracellular pH. The first one is called ammonium chloride prepulse, and the way it works is by addition of ammonium chloride to the perfusion buffer. First, there is an increase in intracellular pH, then it normalizes. And when we take ammonium chloride out, there is actually an intracellular acidification. If we add an inhibitor of sodium proton exchanger, zonipurid, we get an even lower intracellular pH. So these are the investigational tools that we used. And what we saw in a nutshell was that as the intracellular pH decreases, LDH decreases, but PDH actually increases. And this was not as the way we thought it would be, because LDH to PDH ratio should always increase with intracellular acidification. So the explanation that we came up with was that when we add zonipurid to the perfusion medium, we stop NHE1 exchange, so protons cannot get out. So this leads to higher acidification in the intracellular space. But the NHE exchanger is actually located opposite on the membrane of the mitochondrion. So it also stops protons from going into the mitochondria. So the pH gradient actually increases. There are more proton ions outside in the intracellular space than in the mitochondria. And this would increase the activity of the pyruvate carrier because it is dependent on the proton gradient. So this would actually make pyruvate go more into the mitochondria, and therefore PDH would have a higher concentration of the substrate, and the cytosol will have, and LDH will have a lower concentration of the substrate. And this is in line with what we saw, that LDH with zonipurid, LDH decreased and PDH increased. So this is to say that the ratio of LDH and PDH and their activity is not necessarily, is complex. it is dependent on pH, but the ratio is and the response to pharmacologic agents is complex. If I have more time, I can continue. Do I have more time? When am I supposed to finish?
Yes, you have at least 10 minutes more. So of course,
okay,
go on.
So I will continue. I thought it will take me more time, so I will continue. So about ischemia, which is also related to pH because pH decreases, we were asked about what could we say about cerebral stroke? Could we use hyperpolarized pyruvate to detect ischemic penumbra, which is hard to detect and quantify by imaging? Today, with cerebral stroke, with ischemic cerebral stroke, if it's up to 6 hours from stroke onset, thrombectomy is effective. More than 6 hours, the treatment benefit is uncertain. So we thought that if we could add to the clinicians more information about the ischemic penumbra, this could be useful. In vivo models for the ischemic penumbra in rodents are not perfect because the brains are very small and it's the penumbra is heterogeneous. And here our model of brain slices was useful because it provides a homogeneous ischemic penumbra. So this was a system that was useful to study the effects of ischemic stroke and how hyperpolarized pyruvate could detect the state of the cells. So this was the system that we used. Again, we isolate brain slices from a rat, and we used the two systems in order to check the activity of hyperpolarized pyruvate under continuous perfusion or under perfusion arrest. So this is the system of continuous perfusion. When we have the hyperpolarized pyruvate, we inject it through a bypass which goes through to the NMR tube and then washes out. And during this time, we record carbon-13 spectroscopy, as you can see here, lactate production and bicarbonate. And for arrested perfusion, we have this system. So we stop the peristaltic pump. And when we have the pyruvate, we take it into a syringe and directly inject into the NMR tube. And it sits there throughout the measurement. And this is oxygen-poor hyperpolarized medium and creates ischemia. The results were that within a few seconds, we saw an increase in LDH activity, meaning lactate production. And this is the same as we saw with deuterated glucose. But here, of course, the time scale is much faster. With deuterated glucose, we saw this within 2 minutes. But here we see it every 6 or 8 seconds. So this is, of course, faster, but also because the hyperpolarized compounds decay, we can measure for a few minutes and not for hours. So what we saw here is that under ischemia, LDH activity increases and PDH activity decreases. And then we wanted to see what happens during reperfusion, because much of the damage that occurs in stroke occurs during reperfusion. So we wanted to know if we could characterize this damage metabolically. So for this, we used 3 injections. First, a control. The second one, we induced ischemia. And the hyperpolarized agent was given during reperfusion. So immediately the reperfusion was with hyperpolarized medium. And the third injection was used as a control after 1.5 hours to see recovery. So this is a control experiment, and this is a ischemia perfusion experiment. So first, with phosphorus spectroscopy, because we were not measuring carbon-13 during the ischemia, we could measure phosphorus. And when we look at the inorganic phosphate signal under continuous flow and compare it to ischemia, we see the decrease in pH of the tissue. And upon reperfusion, about 1 hour later, we can see that the intracellular pH is back to normal. So this is really tells us that this is a good system for studying ischemia, because this is the definition of ischemia. It's a tissue that is stressed but can recover upon reperfusion. So this is recovery. And the hyperpolarized data looked like this, again, lactate and bicarbonate. And we saw that on the second injection, meaning during reperfusion, PDH activity was lower. LDH activity was actually not significantly different, maybe a little bit higher, but not significant. But the ratio of LDH to PDH was significant, as we saw in the heart during lower pH. So all of these data, they come together and they tell the same story. If I have 2 more minutes, I will tell you a little bit about organoids, which is another study that we initiated as an ex vivo study. So organoids are a powerful tool to understand human diseases. We performed an initial study on brain organoids. And because they are expensive to generate, it is useful to have non-destructive methods for metabolic investigation. And this is what we try to do. But this is a very small piece of tissue. So there was doubt if we will be able to see hyperpolarized signals at all. So we were able to see signals with this system, and here we inject just a dye, and you can see how the dye goes into the NMR tube. You see it darker and darker. It does not go under the filter. The organoid will sit here, and it will experience a homogeneous perfusion medium with hyperpolarized agents. Here you can see it's hard to see, but the organoid is here, and you can also see the organoid is here in the NMR tube. This is a temperature sensor, and this is the oxygen sensor, the organoid, and a Forex filter. So surprisingly, we were able to get phosphorus spectra out of this organoid, and we work with 5.8 Tesla. 10 millimeter bore, so it's not the best detection, but we could get ATP signals to show that the organoid is alive. And we characterized with this video that I showed you, we characterized the we characterized the perfusion characteristics. So when is the food dye there, and we calculated LDH and PDH activities only when the perfusion medium has a constant concentration of hyperpolarized pyruvate. And what we saw is we had 3 injections to the organoids. We did not change conditions because this was just a proof of concept experiment to show that we can see the same LDH activity again and again, and indeed we could. So this is LDH rate in nanomole per second, and you can see that it is constant. It is constant throughout the injection. Even though the signal decays, because we compare it to hyperpolarized pyruvate, we can get the actual rate and it is constant. So for as long as we can detect lactate, the rate of LDH that we measure is constant, and it is constant, or at least similar, between injections. I think maybe I will stop here, and if there are any questions. We'll go to the thank you slides.
Thank you, Rachel, so much for this fantastic presentation. I'm sorry I couldn't be there all the time, but it's a good thing that we recorded it. Thank you so much for being with us tonight. Do we have any questions from the audience? Ken Fishbein.
Hi, this is really exciting work, very interesting and very thorough. I missed the first couple of minutes. Could you explain the rationale for looking at doubly labeled substrates that have both deuterium and carbon?
Yeah, let's go to the first slide. So okay, so the way I organized the talk today is to show you studies on multiple agents, because that's really what we do in the lab. We try to find other agents or research uses for existing agents. So doubly labeled carbon-13 and deuterium is for hyperpolarized glucose or deoxyglucose. So when you, actually I haven't talked about it in this presentation. So if we want to have hyperpolarized carbon-13 that is not in a quaternary position, this is this research, sorry. Let's take it. Go to this. Okay, these are the first studies that we did with DNP. So we wanted to look at compounds like glucose and choline, which don't have quaternary carbons, don't have ketone bodies. And we found that when we deuterate these compounds, the T1 of carbon-13 gets much, much longer. And it gets so long that you could actually visualize it with hyperpolarized MRI.
Ah, okay. This makes good sense.
Yeah, sorry, I didn't talk about it at all today.
So with extensively deuterated compounds, do you begin to see a kinetic isotope effect that would alter the activities that you're measuring?
Come on.
We have not, and there are other groups also who worked on this doubly labeled glucose, and as far as I know, there is no isotopic effect.
Okay.
But it remains to be seen for each molecule and each activity.
Okay, thank you very much.
Thank you.
Okay, Andre, do you have a question?
Yes, I have a question. So maybe like about 10 minutes ago, you showed the slide, and there were a plot there you show LDH and PDH activity as a function of time. And I was just wondering, how did you measure activity with such a high precision? Like it was a few seconds time scale, right?
Yes.
Because you have only one spectrum. I mean, not this graph, but there was some. It was before phosphorus.
Which one?
It was a little bit before organoids. Yeah, it's here.
Before
this
organoids.
one. Next one.
Okay. Okay. Yes, we
I mean,
measure.
Sorry, not here, but it was LD. No. There was a
but I will
LDH
tell you.
activity as a function
I
of
will
time.
answer. I
Here,
will
this
answer
one.
how we,
No, those
sorry,
graph.
no, that's okay. I will answer how we quantify,
Yeah.
because I didn't explain.
Uh,
So the way we work is that we look for the time window that pyruvate concentration is constant in the tube, because, you know, it washes in and it washes out. This is the, in black, you can see the raw pyruvate, sorry, in gray, you can see the raw signal. And in black, you see the corrected pyruvate signal. It's divided in 3. And we look for a time where it's constant. And then we perform a calculation. So we use pyruvate as a standard because we know the concentration in the perfusion medium. This is not easy to do in vivo, but in a test tube with a controlled flow rate and the controlled concentration, because we know how much pyruvate is in the, you know, in the polarization, and we know the dilution and everything. So the concentration of pyruvate, we know. We work with 14 millimolar, and we know the correction factor of the pulse for the excitation of lactate compared to pyruvate. We know the volume of the probe, and we know the repetition time. And in this way, from the signal of lactate and the signal of pyruvate on the excitation of lactate, we get the rate of LDH. And the same is for PDH. So all of it is in this paper, but the actual method is described in another paper, which I can show you. It's called selective excitation. It's by Harris et al. So we first developed this methodology, and then we used it in several preparations.
Interesting that.
Does this answer your question?
Somewhat, but I would think that you should use like first order derivation or something, because you are calculating so you're calculating production of something. So I would expect you calculate the first derivation of the kinetic, but you don't do this. It's interesting.
What do you mean derivation of kinetics?
You have a kinetic, so you need a derivative of it to find the rate of production of something, you know?
No, okay, I didn't explain correctly. What we do is selective excitation, meaning that on each excitation, meaning if we have the spectrum of lactate here, it is fully excited.
Yeah.
The substrate is excited only 5 or 4 degrees, sometimes even 1 degree. This is the meaning of this raw of lactate. From one excitation to the next, this is production. Production within actually 12 seconds. You see there is a lactate pulse, and after 12 seconds, another lactate pulse. So the second one starts from 0.
Yes, because you use 90 degree. Okay, very nice.
Yes. So it's production. It's not the amount of lactate. It's only the signal of hyperpolarized lactate that was formed within those 12 seconds.
Okay,
that's why you don't need derivation. That's why it's a linear thing. Yeah, exactly. This is the beauty of this method. It's not so easy to apply in vivo, although it has been done by Schulte et al. But that's why it's useful, because there is no need for assumptions.
Yeah. Thank you.
Welcome.
Okay, there's another question from Rolf Schulte, please.
Hey, Rachel, very nice work. Very nice overview. Two questions. Can you put for the gadolinium-based alternatives? Can you put a hyperpolarized substrate into a chelate and then inject it? I mean, would that stick if you have like 15N nitrogen? nitrate, 15N nitrate. Can you put that into a chelate and have it injected instead in order to minimize interactions with a exchange to water and so on? Does that make any sense? Or
it makes a lot of sense. I just don't know which chelate would chelate urea or pyruvate, or because if you're thinking about the same chelates as gadolinium, then gadolinium is a positive ion, which is not the case for urea or pyruvate or any other.
Doesn't get trapped inside.
No,
Okay.
because these are the free electrons from nitrogen or oxygen in EDTA and all the compounds. Yeah, but in principle, it's a very useful idea.
Thank you.
Rolf and Rachel, maybe we can add some information on that question. Rolf, you're basically asking, can we add something that prevents relaxation, right? Or reduces the relaxation?
I mean, if you deuterate it and it's exchanging very quickly with water or blood, then you don't gain so much only in the test tube before injection, but after injection, it's very quickly gone. So yes.
Mm-hmm. And there's interesting aspects. There's one paper now from, I'm not sure which group that was, from the States or from Canada, I believe. Maybe it was Chuck Cunningham, who found that the T1 of pyruvate was prolonged even in vivo if you just do it with D2O in solution.
True.
Okay, that's very interesting, I find. So it sort of sticks to the pyruvate or does something else. And maybe Andre or Josh want to comment on the cider effect briefly, without stealing Josh, Rachel, the show.
Maybe I could add something until they answer. Actually, Rolf, within the Alternative to GAD project, we also try to encapsulate urea and other compounds and encapsulate it with D2O. It turned out to be extremely hard to encapsulate water. So we used all kinds of nanostructures, nano capsules, and water always escaped. Eventually, we worked with the group in Max Planck Institute for Polymer Research. And actually, they came up with a very interesting idea towards the end of the project, but it has to be proven yet. But by conventional methods, we saw that it's impossible to lock water or D2O. Yeah.
Interesting. And one final question there. Have you looked at permethylated 15N glutamine? I mean, that is also a perfusion agent.
Yes, no, we have not. We have not. But the question is also about safety. I'm not aware enough. I haven't looked at the safety profile of this compound, but the compounds that I have shown you are extremely safe and extremely soluble in water. So this is another aspect that should be considered. And stable.
Thank you. Thank you.
Thank you.
All right. Do we have any other questions from the audience? Ken, again, please.
Yes, thank you. So the work on ischemia reperfusion makes me think of some work that we did many years ago that relates to an interesting protective effect, where if you have numerous brief episodes of ischemia, and then you have a longer episode of ischemia, that you get less damage, that there's actually less loss of cardiomyocytes and so forth. So this is something that might be interesting to evaluate. This is actually was motivated by some clinical observations of people who have had, for example, transient ischemic attacks, or who've had episodes of brief infarction followed by a longer one. They seem to do better. So
So I'm actually extremely interested in this, and you have to excuse my ignorance. If you could send me this work, it's really interesting because we saw something similar with our work on the deuterated glucose in ischemia, because we could observe for a long time. So we actually, in one experiment, we did a third ischemic period, and we actually saw the cells dying, and then after reperfusion, they came back after 1 hour. So
Yeah.
this is something that is, I couldn't figure it out, but it's an observation. So it's good to know that that it has been observed before. So it's very
it's
interesting.
counterintuitive, I know, but
Yes.
yeah, I'll be
Yes.
happy to send that to you.
Thank you. Thank
You're
you. I
welcome.
appreciate it. Yeah.
All right. Do we have any other questions? If the hands don't go up, or we wait a little bit, we have 3 minutes till 7. I'm just commenting on Rolf's comment or question. So what we found is if we add some molecules to, for example, N15 nicotinamide or urea, we get a much increase of T1 at low fields. So a very, very strong increase in T1, like double or triple. We think at lower fields even more. We could only measure down to 0.5 Tesla. And we believe that that works by the molecules making hydrogen bonds with the exchanging or the exposed ring nitrogen. I'm sorry, I'm herding 5 kids at the same time. That makes it a little bit complex. Andre, do you want to comment on?
Yes, I have a question about the last section you presented about these organoids. Can you show this nice
Yes, of course,
in the mud tubes? You developed the.
Of course. Is this what you?
Yes, this one. How did you machine or did you make this holder for the organoids?
The filter?
Oh, yeah, or filter is it? Okay.
It's custom made by the company called Porex.
And
that's why it's specifically made by dimensions that I gave them. And if you're interested, I still have some.
And this is 10 millimeter in the mud tube, right?
Exactly. So there's 10 it has the inner diameter of the NMR tube, and it has a hole inside, and actually it looks like this is the perfusion, but it's not. It's just a line that holds it in place, you see? So this is the same line as the perfusion because it's made of PEEK, so PEEK. So it has the no susceptibility interference, but it's just to hold it in place. And then you have the perfusion somewhere here and the sensors. So there's another line for perfusion, and one line was just used to hold it in place. So it was not connected to perfusion, actually.
Yes, yes. And this oxygen sensor, I saw once an oxygen sensor, and they need a kind of marker on the wall of the tube. Does your sensor needs anything like this?
What do you mean a marker?
I don't know, like a sticker.
Yes, there is a sticker. There is here. You can see it. You see, this is the sticker.
Okay.
See?
Yes.
Oh, I was going to say, I know exactly what you mean. There's a company, Presense, makes these fiber optic sensors, and they give you 2 options. One of them is a spot that has a fluorophore that's oxygen sensitive, and you can simply put it on the inside of your vessel and then shine light through the wall of the vessel. So you have a completely non-invasive measurement of oxygen. They also do pH and carbon dioxide, I believe, or you can have a fiber optic with the fluorophore on the end of the fiber like this.
And here you don't stick it to the wall, you just stick to the sensor, right?
You always stick it to the sensor.
And then you change it each time.
It has to be, you have to stick it at the end of this fiber optic. Yeah.
Oh, nice.
And it's useful for a few measurements, and then you have to replace.
And how do you know it's not working anymore?
We always calibrate before the ex vivo measurement. We calibrate, with buffers and in the spectrometer, outside the spectrometer, we compare between two, so you develop an experience.
All right.
Yeah.
And then you show that you measured it.
Maybe my student could answer this question better, but this is my answer.
Right. You measured organoids 4 times. Was it 4 different organoids or the same organoids you measured 4 times? I'm just curious.
For the same organoid. We measured several, we measured the 3 samples, so 3 different organoid samples. Here you have just one that was actually big. The other samples were 2 or 3 smaller organoids that also sat on the same Borex filter.
And then you injected 4 times to the same organoid, the hyperpolarized part of it? Or?
3 times.
3 times. So you take it out, wash it, and then...
No. It's in the NMR tube. The perfusion continues. And we have the continuous flow that I showed you here.
Okay.
So when we have the hyperpolarized sample ready, we inject it.
And the continuous perfusion of media.
Yes. So the continuous the bypass is only for the hyperpolarized medium. So the pump continuously perfuses the slices. And only when we have the hyperpolarized medium, we inject it to the bypass, change the connection of 3-way valves. So the pump pushes the medium through the bypass and into the NMR tube. And when it's finished, we go back to the regular flow. So that's how you can do 3 injections to the same sample without taking it out.
And then perfusion media does not mix much with the hyperpolarized media. Then.
We can control it. Sorry, we try not to mix it because while we have this, while we have this flowing with the bypass, we will not return. You see, there is an outflow. So we take the outflow line outside and let it flow outside the regular medium. So we don't build up too much pyruvate inside or nothing at all. We don't want it perfusing. We don't want the hyperpolarized medium to accumulate here. So we just take the inflow line out until everything is measured, the volumes are measured, so we know how much to, and the time is, and the flow rate is constant, so we know exactly how long we have to keep the outflow outside so it doesn't mix. I have to tell you, it's not 100%. So, but that's what we try to do. Sometimes we do get some mixing, and we see it. If we have it, then we will see it with the increase in phosphorus, because the buffer that we used had 50 millimolar of phosphate buffer. So if we do mix it, we see it in the phosphorus spectrum.
Okay, thank you very much.
Welcome. And I wanted to tell you something more about the sticker. You see, here it's very close to the organoids, but with the brain slices, we had to keep it a bit up because the slices would stick to it. So it's not a perfect, but it's better than nothing. And still, it gives you something constant at a constant height that measures oxygen and temperature. Yeah.
All right, Rachel, thank you. And thanks all for this interactive discussion. This is really how I envisioned as well the seminar. It's a discussion about the things that interest us. So last chance for anyone. Pick up the microphone and ask the last question to Rachel, our speaker tonight. That doesn't appear to be the case. So, Rachel, thank you so much for being with us.
Thank you
if
for
you don't
having
mind,
me.
yes, anytime.